Hello and welcome. My name's Margaret, and today I'm going to go over some media preparation for hemp tissue culture with you, and also show you some explant preparation for micropropagation. And these are pretty standard processes for tissue culture in general, so if you're interested in tissue culture, this is a great example. So we're going to start off with our autoclave portion of the media. This contains some activated charcoal and agar. So the agar is the gelling agent to solidify the media to provide a structure for the plants to grow in. And the activated charcoal helps to prevent polyphenol oxidation or browning, so it helps keep the plants from getting brown and mushy. So these are going into 500 mils of sterile water, and this half of the solution is going to be autoclaved to sterilize it. So I'm just swirling that up to mix it, and then I'm going to adjust the pH to about 5.7 plus or minus 0.05, and that's just the optimum pH for plant growth in this case. So this solution starts off basic. I added some hydrochloric acid to bring that down, and this pH adjustment can be one of the most time-consuming parts of the media making process. So I only showed you a tiny part, but this can take a while, just adding a couple drops at a time to get that pH right in the desired range, and it can take a while to stabilize. And once that's done, it's going to go into the autoclave to be sterilized. So here it is, finally evening out on that pH meter. There it is. And then the lid's going to go on, and we are headed to the autoclave. I also autoclaved some magenta boxes at this time. So a good rule of thumb for the autoclave is about 10 minutes per liter of solution. So here we're in a wet cycle because this is we have liquids in our autoclave. And yep, so I did 60 minutes because I had about 6 liters of solution in there, and then that's going to go in the autoclave. So while that's in there, we're going to make our filtrate, and the filtered portion contains both a basal medium and a carbon source, and also in this case some metatopolin, which is a plant growth regulator. So all of these components are going into 500 mils of sterile water. I've already added different combinations of the media and the carbon source or sugars, which is just food for the plant, because that's the experiment that these are for. And then I also included some ppm, which is particularly important in hemp tissue culture to prevent contamination. And then again, we're just adjusting the pH to about 5.7. In this case, the solution is more acidic, so we have to add some sodium hydroxide to bring that pH up to about 5.7. Now, these components are not stable at high temperatures, so they cannot be autoclaved for sterilization, and that's why this half of the solution we do separately. So to sterilize it, I'm in the laminar flow hood. This is all sterile. This bottle that I'm pouring into has been autoclaved, and this is a sterilized filter, and it's hooked up to a vacuum. So as I pour in the solution and it goes through the vacuum, it's been sterilized. And this bottle has already been sterilized, so we can add that to the autoclave portion. And this is sort of time sensitive because agar sets up at a certain temperature, so we have to work quickly. I'm just combining the autoclave portion, which is cool to the touch, so it's not going to degrade those heat-sensitive elements, but it's still hot enough so that the agar hasn't solidified yet. Just give those an extra good shake, and then those can be poured straight into sterilized magenta boxes. Now, normally I would do 20 per liter because about 50 milliliters of solution go into a magenta box, but I was a little short on magenta boxes this time. So normally they'd be about half that full. And you can see it's important to work quickly because the agar can set up very quickly as it cools. So just getting those into every magenta box And again, we're in a laminar flow hood, so this is all sterile. Everything's been wiped down with isopropanol to keep things sterile before entry into the hood. And you can see that there is some bubbles, so I'm just popping those with some tweezers, which I've sterilized in the microbead sterilizer. Those bubbles can just interfere with the structure, and we don't want anything messing with our plants. So it's important to pop those for a smooth surface. And these have to set for a while before we can put the lid on. So once they're solidified, we can go ahead and put that lid on, and we're ready for the next step, which is explant preparation. So now that we've made our media, we are ready to get our plants ready. So I've already taken some cuttings, and I'll show you what they look like here in a minute. But first, we're going to surface sterilize them with some ethanol. And these are just in a tea strainer, which is really convenient for dunking plant parts in different solutions. So those just get a quick bath in ethanol for 1 minute to surface sterilize, remove any contaminants, and then they're going into a solution of sterile water and a little bit of that same ppm just to rehydrate, and that's for 30 minutes. After that, they're going to get 10 minutes in the solution I'm making here, which is 3 parts water, one part just regular bleach, and a splash of tween, which is detergent. So this is our last sterilization before the plants are going to go into tissue culture. And this is just to ensure that they are completely contaminant free, especially since hemp is particularly prone to contamination in tissue culture. So I'm just gonna mix that up, and then that stays for 10 minutes before we get our plants into magenta boxes. There they are. And then I'm just going to rinse thoroughly with sterile water, and that's to ensure that there's no bleach or detergent residue on the plants, which could potentially mess with them in tissue culture. Just being super thorough here, rinsing with some sterile water. There it is. Plants are very clean. And we're going to dump those out into a fresh petri dish. Once again, we're in the laminar flow hood. Everything has been sprayed down with isopropanol before entering. You can see our little cuttings there. So these are just axillary buds that have been taken before the axillary bud has started to come out. So here's my tools being sterilized in the microbead sterilizer. Those sit in there for about 30 seconds, and it's really important to cool them down before using them on plants because they could obviously burn and damage the plant material, and then the plants would not grow like we want them to. So those have cooled, and then I'm using my scalpel here to cut off the very bottom of the bud, which you can see has some oxidation and damage from the sterilization process. So we want to make sure that the stem is easily able to imbibe the nutrients. And then the stem just goes straight into the media so that the Y of the bud is sticking out right at the media surface. And again, these are axillary buds cut so that the axillary node has not yet produced leaves, and it's ideally just meristematic tissue, and that allows it to grow here in the tissue culture into vegetative growth. So yep, that one went a little far and just poking it back out there. And usually about I do 4 plants per magenta box. So they go in, and then once they're in, we'll put the lid on and then seal that lid with grafting tape so that no external contaminants can enter the box. And then they will go into the growth chamber, and here are some plants after a while in the growth chamber. You can see there's light on all sides, and this is what the plants look like after about a month in the growth chamber, depending on the media. So you can see they're getting a good start in life, and then from here, the plants could be subcultured, or you'd have to move media because nutrients only last so long, or they could even be transplanted into the greenhouse. So either way, the plants live happily ever after. Thank you for watching and learning all about hemp tissue culture. The end.